So my name's Tom Zoeller. I'm an endocrinologist at the University of Massachusetts at Amherst. And I was really trained as a basic endocrinologist. I did kind of molecular neurobiology, neuroendocrinology at the NIH in the 80s. And my real focus is thyroid hormone and brain development. And in the mid-90s, I was asked to be on the Endocrine Disrupting Screening and Testing Advisory Committee that was set up by EPA. I have no idea why they asked me. It kind of came out of the blue. I thought it might be fun to come to DC. It was about a year and a half. It wasn't fun. But when I got finished, I realized that the way we're approaching, the way the system is approaching this issue is not going to be effective because they're not using basic principles of endocrinology that I have been trained with really my whole career. So next. So what I want to start with is to make really two propositions at the beginning. The first is that human health is being affected by endocrine disruptors today. And I'll show at least part of the evidence, certainly not all of it, but part of the evidence that I think I can defend that statement scientifically. Secondly, the safety system that's designed to identify endocrine disruptors and essentially weed those out or at least have appropriate risk assessment simply isn't adequate. Next. So first of all, what's an endocrine disruptor? I come at this from an endocrine point of view. I've been a member of the Endocrine Society since the 1980s. I really am sort of, I think in terms of mechanisms of hormone action. So there are many different definitions that you can see from the US EPA or the World Health Organization. The bottom line really is that an endocrine disrupting chemical interferes with hormone signaling. In some way, hormone signaling is very complicated. You can imagine that there are different chemicals that interfere with the system at different places. So let me just make one point. I don't think it's possible to interfere with hormone action during development and not have an adverse outcome. So adverse outcome is part of the definition, but I don't think that you can interfere with hormone action during development and things turn out okay. Next slide. So what are the implications of this in terms of the USGS studies in terms of humans? Well, because fish aren't humans, what difference does it really make? Next. First thing is that these hormones that we're talking about, estrogen, testosterone, thyroid hormone, glucocorticoids, are chemically identical in fish and in humans. So there's absolutely chemically, there's no difference between a human estrogen and a fish estrogen. Next. Hormones act on receptors. If you don't have a receptor, you don't have any hormone action. So it's the protein that the hormone binds to that actually does the work. Therefore, I'm showing in this left panel here, I'm showing a hormone receptor interacting with DNA. These hormones regulate gene expression. Estrogen, testosterone, thyroid hormone, that's what they do. They regulate gene expression. They turn them on, they turn them off. When the hormone binds to the protein, to this receptor, it's kind of like a key and a lock. The key is the hormone, the lock then turns and does something. It activates gene expression or it turns gene expression off. If the chemical binds to that lock, either it activates inappropriately that receptor function or it blocks the action of that receptor. It's more complicated than that, but that's a good way to start to think about it. Next. So, of all the things I can talk about, I should kind of be talking about reproduction because that's what you just heard about in fish. I'm really a thyroid guy and that's what I know the best. I know this literature pretty well, so that's what I'm going to focus on. And I think it's also germane to this topic. Next. So first of all, thyroid hormone is essential for brain development. Every baby born in this country has thyroid function measured at birth because if thyroid hormone levels are low, the effects could be disastrous. And you can see two babies here. The one on the right has congenital hypothyroidism, which is technically an idiopathic deficiency in thyroid hormone. You don't necessarily know why it happened. If you can identify those kids early and give them thyroid hormone, then they can reach kind of normal stature. As a population, they have continuing deficits. Thyroid hormone acts at different times during development. This is kind of an important issue. Obviously, if thyroid hormone or if thyroid function or thyroid hormone action is disrupted during this period, there's going to be adverse effects. So that's one of the main reasons that I look at this. Next. So, in fact, there are hundreds of chemicals in the environment that can interfere with thyroid function or thyroid hormone action. The fetus doesn't begin to produce thyroid hormone until about 12 weeks of gestation, and doesn't really make enough for itself until about 20 weeks. So maternal thyroid function is critically important during pregnancy. Next slide. I think one of the best literatures about chemicals interfering with hormones that impact cognitive function in children actually was done here in DC with PCBs, polychlorinated biphenyls. We don't think about a lot because their production was banned in the 1970s, but we're all still exposed, and we kind of make the argument that we haven't learned our lessons because we continue to make chemicals that are similar to this. These chemicals can bind to the thyroid hormone receptor. We know from a large number of studies in animals and biochemical studies, they increase the incidence in animals of behaviors that look like attention deficit. They're also correlated with attentional problems as well as low IQ in people, and one of the greatest predictors of that is this structure in the brain from A to B is the corpus callosum, which is critically dependent upon thyroid hormone action. So here we've got a brain structure that's dependent upon the hormone that we know PCBs can interfere with. This literature is very large, and I think produces a very important statement that endocrine disruptors in the environment can interfere with hormone action and produce adverse outcome in kids. PCBs are a large class of chemicals. It's clearly not just acting as an endocrine disruptor or a thyroid disruptor, but probably has several other specific effects. Next. We can see that, at least today, 1 in 6 children in this country below the age of 17, I think from 3 to 17, are diagnosed with a neurobehavioral, a learning disability, or a neurobehavioral problem like ADHD. 1 in 6, so almost 20% of children today are diagnosed. There are many arguments about what the cause of this is, whether it's increasing. It's a complicated issue, but I don't think that we can ignore it. Next. Autism also, the incidence of autism prevalence is about 1 in 110. Of course, this is gender skewed, and so about 1 in 58 boys are affected with autistic spectrum disorder. Thyroid hormone is clearly, at least speculated, to be involved in the etiology of both of these. It's certainly not the only factor, but it is a factor that probably contributes to both of these. Next slide. So, are Americans exposed to EDCs? That's the next kind of comment. Now, the CDC has a biomonitoring program that they apply to adults, but they don't apply to the fetus. So there's no government agency that is doing biomonitoring in fetal fluids or tissues, and I think that'd be a great idea. The few studies that have been done that are kind of global have identified, in this study here, in 10 samples of cord blood, they identified 287 different chemicals. About 40-something chemicals were the same in all 10 of those babies. That's a huge chemical burden. At the same time, there are a number of studies all over the world that have been looking at PCBs, flame retardants, bisphenol A, pesticides in amniotic fluid, in cord blood, and breast milk. It's clear that all of these fluids are contaminated with a large number of chemicals, many of these at concentrations that in the lab I can show interfere with hormone signaling in animals. Next slide. So, few of the chemicals, and I'm kind of transitioning now to talking about the safety system that's really set up to identify this kind of chemical. The U.S. either produces or imports an estimated 73 billion pounds of chemicals per day. There are 80,000 chemicals licensed. About 3,000 are produced at amounts of over 1 million pounds per year. So this is a high production volume chemical. Very few of these have been evaluated for these kinds of activities. Next. So I'm going to make the argument that the system designed to protect us really isn't working. And there's a couple of main points that I want to make. The first is that the kind of standard way of looking at toxicology, which is a valid and valuable way of looking at things, really doesn't work for hormones because the way hormones act. Next slide. So in 2009, the Endocrine Society put together kind of a large compendium. In fact, that this is, I guess, outside. This document is outside. I think it's got 400 and something references. It's a very solid scientific statement from the Endocrine Society that really concludes that endocrine disruptors in the environment are a significant issue that we need to deal with. And we need to deal with it appropriately, not by using methods that are really too old or not appropriate for endocrine activity. I'm involved in a task force that is beginning to take this a little bit further and talk about what kind of endocrine principles should we be using to identify endocrine disruptors. Next slide. So on the left in this panel is the Tier 1 of the US EPA's endocrine disruptor screening program. So this is kind of the first line of defense to identify chemicals that are endocrine disruptor. If a chemical goes through this and is deemed to be not an endocrine disruptor, then they don't say that it's that it's okay, that it's safe. What they say is that it's not going to be further studied because there are other chemicals that might be more important to look at. The argument that I'm going to make is that this kind of program really isn't going to work. Next slide. So the first principle about hormones is they act on receptors. The key issue about receptors is there's a limited number. So what this means is that you have a dose response that looks like this S-shaped curve. The critical issue there is that it's saturable. You keep increasing the dose of the hormone, and at some point you've used up all the receptors, and therefore increasing the hormone doesn't produce a bigger response. Next slide. In contrast, if you think about a traditional kind of, not traditional, but a toxin, one that doesn't act through a receptor, and the one that I kind of think of is alcohol. Okay, so you start drinking alcohol, nobody thinks that you will reach a place, say at 0.08, a blood alcohol concentration of 0.08, where you can just drink forever and it will never make any difference. That's the way hormones act. You can keep loading the hormone on and it will not produce an additional effect. But for other kinds of chemicals that don't act through a receptor, it's a linear dose response that ends in something bad, like death. Next slide. In the endocrine disruptor screening program, essentially what they would do is use 4 doses. And what they're looking for is in the black bar here. So you get a stair-step effect that's kind of a linear or reflective of a linear dose response, where a hormone active or an endocrine disruptor may produce an effect that looks more like a square wave because the lowest dose that they use is already over the saturation point for the receptors. This is actually incredibly common. I've read a number of risk assessments. I'm on the Science Advisory Board of the EPA, and we have to go through all these IRIS assessments, et cetera, et cetera. So I've looked at a lot of these, and it's amazing how many times this dose response shows up and is discarded as being not dose dependent. We cannot do this. So we have to use endocrine principles both to design our assay, but also to interpret the results. Next slide. In addition, hormone receptors can be downregulated. So a high dose of a hormone can cause the receptor to actually disappear. So you get this weird high dose. In fact, the hormone can actually cause its receptor to be downregulated. This is actually the basis for some pharmacotherapies, for example, for prostate cancer. So this is so well known in the field of endocrinology that it's actually used in clinical medicine. And yet, this is an issue that is completely discarded in the endocrine disruptor screening program. Next slide. receptors, this is actually a picture of thyroid hormone receptors early in development. This is a really fancy kind of molecular technique that these guys used to show this blue color wherever thyroid hormone receptor is activated. You can see the nervous system here. A and B represent one developmental stage. C represents a later stage. You can see how the blue color is spreading. What that means is that the receptor is present at different places at different times during development, and it's being activated. For example, if you look at the eye in the panel A early in development, you don't see any blue color, but you do later, which means that the receptor became expressed and then it became activated. So what this is telling us is that during development, receptors for hormones exhibit a very specific spatial pattern of expression, and that changes over development. If you do a long-term study and don't take this into consideration, you're going to miss things. Next slide. In addition, many hormone systems have multiple receptors. There are 2 receptors at least, estrogen, ER, alpha and beta, maybe gamma. There's also speculation about membrane receptors. These are important kind of molecular kind of signaling pathways that people are studying. Thyroid hormone receptor has a number of classes of receptors, 2 different genes, multiple kind of proteins that mediate this. Also progesterone receptors as well as adrenal hormones have multiple receptors. Next slide. So if we go back here, I can actually point to different places that have different receptors. So what if you have an endocrine disruptor, and we've studied some of these, that will bind to the alpha receptor but not the beta, or the beta and not the alpha. All of a sudden you have a mosaic of effects that don't recapitulate a traditional endocrine disease. And that shows up poorly in these kind of standard assays, and it shows up poorly even in population studies. So we have to be very careful about the complexity of endocrine systems and how this is playing. Next slide. Oh, it is the next slide.
I was a little ahead of you.
So the point that I'm trying to make here is that the chemical will be identified as an endocrine disruptor if it binds to, let's say, the ER alpha, because that's what makes the uterus get bigger in this uterotrophic assay that they talk about, or if it produces this kind of standard effect. If it's not standard, it won't show up. Also for the androgen, and the same with thyroid hormone. In all of these cases, in fact, thyroid hormone signaling really isn't evaluated in either Tier 1 or Tier 2 of the endocrine disruptors screening program. And why is it that every baby born in this country is evaluated for thyroid function, but it's not represented in the endocrine disruptor screening program? It makes no sense to me. Next slide. So these things are going to miss. This endocrine disruptor screening program is going to miss chemicals, for example, bisphenol A, that binds to the ER beta, or that has sort of what I'm calling non-canonical kinds of activities. There's a couple of papers showing that bisphenol A is equipotent to estradiol in the pancreatic beta cells. Now, we don't really understand how this can happen, because if we pull the receptor out and stick it in a test tube, it doesn't bind to bisphenol A very well. But of course, the estrogen receptor doesn't live in a test tube. We don't really know the constituents of the beta cell that sort of modifies the three-dimensional structure of the estrogen receptor. So, next slide. All of these chemicals here, we've studied actually in our lab and have found that they bind to thyroid hormone receptor and produce effects that are very different from what thyroid hormone itself does, both in the animal as well as kind of the molecular kind of biochemical effects of these chemicals on thyroid hormone receptor. Really, I would imagine that everybody in this room is contaminated with each of these. Certainly bisphenol A, I think CDC has demonstrated that 93% of adult tissue samples, these are spot samples, contain bisphenol A. PCBs, everybody has. Polybrominated diphenyl ethers, if you were going to make a chemical looks like thyroid hormone. That would be one of them. Triclosan would be the other. Triclosan also is coming from places that I don't think we fully understand because people are much more exposed than you would kind of predict. Next slide. So, in conclusion, to just kind of summarize this, first of all, chemical exposures in the human population are pervasive. This safety system that we've tried to develop, and I kind of want to make 2 points about this. First of all, I don't think it's adequate. Secondly, this is a very difficult thing, and I think EPA is doing the best that they can do with what they have to work with at the moment. They need to recruit experts from many different medical societies, in my opinion. Same with FDA. They don't always share that opinion, but it is a tremendously difficult issue that they're up against, to protect public health against the number of chemicals that we're all exposed to. So I'm not trying to trivialize the difficulty or to lay too much blame on EPA, but it's not adequate, and we shouldn't turn away from that concept. The application of endocrine principles can improve the system the way it stands, but it also needs to improve a design of a new kind of generation of assays. As we do these risk assessments, we have to incorporate modern science. We cannot continue to make the argument that GLP defines good science. It simply doesn't. We're ignoring the science that essentially makes this one of the best healthcare systems in the world. We're ignoring that so that we can go to GLP to protect public health from these kind of chemicals. That makes no sense to me. So I think I'll...